Yeah, but they answered my question.
I have one question.
Keep the rest of the questions for the coffee break. I have a couple. Our next speaker is Ángeles Almeida of the Instituto de Investigación Biomédica de Salamanca, here in Salamanca. And she's going to talk about the P53 arginine 72 proline polymorphism determines cognitive decline progression in Alzheimer's disease.
Okay. First, thank you very much to Cebernet and also Fundación CIEN for giving me the opportunity to share with you our recent results. And it is a big pleasure to be here in this fantastic meeting, and especially because it is in Salamanca, my city, so I'm really very happy to participate on this. I'm going to talk about the effect of P53 on cognitive decline progression in Alzheimer's disease. So thanks to the fantastic talk of Professor Sud of yesterday, so I can pass through the introduction probably faster, and then we have time for questions. So as you know, Alzheimer's disease is the main cause of dementia, and it is affecting around 50 million people in the world. So because of the aging of the population, this prevalence will increase. So that represents a very important sanitary problem. But also, Alzheimer's disease represents 1 every 3 deaths in the elder life, and it is estimated to cost around more than 1 billion euros per year. So for all these numbers, all these reasons, Alzheimer's disease is probably one of the most lethal, expensive, and devastating diseases of this century. I show you several warning signs of Alzheimer's disease. The most frequent are memory loss, problems with languages, and also anxiety and changes in mood and behavior. And all these Alzheimer's disease signs are related to parallel changes in the structural brain of the patient over the time. So at the beginning, there is a loss of the connection between the neurons. There is a synapse loss. And at a later stage of the disease, there is a shrinkage of the cortex and also the hippocampus and enlargement on the ventricles. And all these changes are caused because of cellular and molecular changes in the brain. So as you know, the two hallmarks of Alzheimer's disease is the accumulation of amyloid beta. The amyloid beta accumulates in plaques in the extracellular outside the neurons. And also there is a phosphorylation of tau that forms intracellular neurofibrillar tangles. So these two hallmarks are quite related to the disease progression. And what is very important is the fact that even when the tapes, when there is a cognitive decline in the patient, around 10 or 15 years before, there's an accumulation of amyloid beta. Then this accumulation of A beta is one of the first symptoms of, well, it's not a symptom, but it's an event in the disease. Later, there is a phosphorylation of tau, which is very related to the onset of neurodegeneration, as you can see here, a functional deficit at the later symptom that appear in this. So there is a clear difference in the age, and even with normal cognition, there is an accumulation of amyloid beta. As well, you can see here that also the neurons are affected, or other cells. Also, there is a dysfunction of the microglia, and also there is reactive astrocyte. But neurons are more vulnerable to amyloid beta. And the fact that many clinical, very intensive effort has been done to develop drugs to stop Alzheimer's disease progression, and mainly are focused on the targeting this amyloid oligomers, which are considered the most neurotoxic compounds of amyloid beta. But most of these clinical trials, as Professor Sudov explained fantastic yesterday, has failed. And this also is probably due because there is an equilibrium between of these species, the monomers and the plaque. So probably it is necessary to target not only in one step, but also in parallel in different steps. In any case, the result is the damaged neurons. So we have been, for a long time, interested in studying the molecular mechanism involving neuronal death with the aim of establish a new molecular target to develop new therapies, but also to identify biomarkers of disease progression. And if we think in neuronal death, so p53 has a very important role. p53, as you know, is not necessary to introduce it because it's probably the most studied protein and is implicated in very, very important processes, senescence, apoptosis, cell cycle metabolism. And it is quite well implicated also in neuronal death. So because p53 is implicated in all these important processes, it's very well regulated. And the main intracellular regulator of p53 is the E3 ubiquitin ligase MGM2. So MGM2 polyubiquitinase p53 and target it's for proteasomal degradation, but also monoubiquitinase the protein and induce the export of p53 from the nucleus to the cytosol, just prevented the transcriptional activity. In turns, p53 induced the expression of MDM2, and then there is a quite nice loop between them in regulation. But under stress conditions, p53 suffered many post-transcriptional modification, and one of the main studies is the phosphorylation. So this phosphorylation in p53 prevents the interaction with the ligase, and then there is an accumulation of the protein. And this accumulation, the protein goes to the nucleus to activate apoptosis by activating pro-apoptotic genes, but also interact with different proteins of the BCL2 family proteins, interact with the mitochondria. And as I show you during this talk is that the different interaction of p53 with either the mitochondria and the nucleus will regulate the balance between neuronal death and survival. So this crosstalk between the mitochondria and the nucleus of p53 is very important for neurodegeneration. We first wanted to know whether this mechanism, whether p53 has really an important role in Alzheimer's. So as a fresh approach, we use cortical neurons, we incubate with oligomerized amyloid, and we measure apoptosis by measuring caspase reactivity. And as you can see here, there is an increase of the activation of the caspase, but this activation was prevented with when the p50, with an inhibitor of p53 and also with roscovitin, that's phosphorylate that inhibit CDK5, which is phosphorylating p53. So interestingly, we found that those neurons, dying neurons, were those what accumulate p53. So we describe the total signaling pathway and with different experiments, and I don't show you because of the time, but we found that amyloid beta increase calcium, and this calcium promotes the activation of CDK5. It's a CDK quite predominant abundant in neurons, and this promotes the phosphorylation of p53, then inducing mitochondrial damage and also apoptosis. So it seems that p53 was involved in amyloid beta neurotoxicity. So our next step was doing it in vivo, And we injected intracerebroventricularly oligomerized amyloid beta in wild type and also in p53 cow mice. And we found, well, as you, this is the accumulation of p53 in the neurons in the hippocampus, and we found an important dendritic disruption induced by A beta, and this effect was not observed when in the cow mice. So it seems that p53 was really also in vitro affecting neuronal death. And this was confirmed by tunnel, as you can see here. There is in the hippocampus tunnel positive neurons induced by A beta, but we couldn't find this in the, voy a quitar esto, in the cow mice. So yes, we could demonstrate that amyloid beta was promoting neuronal death in the hippocampus in a p53 dependent manner. This is in the protein, in p53, there is an important domain, the proline-rich domain, which is involved in the regulation of pro-apoptotic activity. And it is very interesting because in the codon 72 that is encoded this region, there is a functional polymorphism specific for humans that results in the substitution of proline to arginine. So this change of these two amino acids in this proline-rich domain changed the electrophoretic activity of the protein and also promote conformational changes that condition the apoptotic role of p53. So in tumor cells was demonstrated that the arginine p53 were more pro-apoptotic than the proline one. So we decided to study what if this polymorphism was really affecting the neurotoxicity of amyloid beta. So as a fresh approach, we use again cortical neurons, and we transfected cells with the two polymorphic variant, proline or arginine. And as you can see here, in the case of the arginine variant, the neurons die in a dose-dependent manner. However, the proline were quite healthy. So something was happening and something was affecting. So the polymorphism really may affect the different activity, the pro-apoptotic activity of p53. And we studied why. And we couldn't find any difference in p53 stabilization or even all the targets. We tried p21 and also other pro-apoptotic targets of p53. So we discard that was due to a transcriptional activity. But what was very interesting is that when we look at the localization of p53, we found that the arginine p53 was mainly located in the mitochondria, but this was not happening with the proline one. So we identify a different subcellular localization of these two variants. So we did more experiments, and I show you here only the immunocytochemistry. I don't know what happened here, but, and we found that the arginine interacts with BCLX. So the fact what was happening is that the p53 arginine variant can go to the nucleus and also can interact with BCLXL in the mitochondria. So what it was to inactivate BCLX, so that promotes the dimer between bags and back, and then promote the release of cytochrome C and apoptosis. So in contrast, proline can also interact with the mitochondria, but in a much lesser extent than arginine. So the fact that only goes to the nucleus that makes it less toxic for the neurons. Because this is a human-specific polymorphism, it's not expressed in mouse. So we use knocking mouse expressing these two, either the proline and arginine, and we did also neurons, we incubate with oligomers, and we found that both proline arginine accumulates here after exposure with amyloid beta. And this was, well, we identify that also there is a phosphorylation, but as you can see here, the accumulation of TCASP3 was higher in the arginine neurons. So this was confirmed by immunocytochemistry. So neurons harboring the arginine variant are more vulnerable, more susceptible to amyloid beta-induced neurotoxicity than the proline one. We investigate, we identify the molecular mechanism, and if we look at the cascade we explained you before, we found that there were no difference in calcium release. It's exactly the same with the two variants, and also the activation of p53. We found the same more or less in both genotypes. Then we go further and we go to the mitochondria, and we found that again, in contrast to proline, arginine after amyloid beta exposure of the neurons accumulates in the mitochondria. We also study the mitochondrial respiratory rate of these neurons, and we found that under basal condition, the control, sorry, this is the basal condition and this is the maximum after stimulation, maximum respiration, this is the seahorse. So before amyloid beta exposure, the respiration was the similar. However, we found that in the arginine neurons, amyloid beta decreases the maximum at the basal, as you can see, here is the number, respiration as early as 4 hours, and this effect was not found with the proline. And even at 24 hours, we found an important decrease that was not found in the proline. So we detected, we identified that both neurons, arginine and proline, respond in a different way to amyloid beta, and the mitochondria was implicated in this different response. Here I show you that this is only to summarize these results, that the traffic between the nucleus and the mitochondria of p53 is essential to regulate amyloid beta neurotoxicity. So it's more toxic when p53 interacts with the mitochondria. It's more toxic and it's quicker, the effect. Next, we do this experiment in vivo. This is with the knock-in again. We injected oligomerized amyloid beta, but what we did also was to inject a p50mu that inhibits the interaction of p53 with the mitochondria. So in this case, we found that this is arginine. As you can see here, there is a tunnel, positive cell. There is a higher dendrite disruption and a higher neurodegeneration. But in both cases, this was prevented when with p50mu. Then with this drug, we transformed the arginine mice to proline mice, that in this condition, all animals respond at the same way. Here is just the novel object recognition that, as you know, is a learning and memory test. So as you can see here, the arginine has a cognitive decline here in relation to proline, as with p50mu, we could restore quite nicely the cognitive decline of animals. And this was also confirmed with the open field that, as you see, is a test that could measure anxiety in animals. So as you can see here, the arginine treated with amyloid beta is is all the time in the border, so it's not going to the center. That means that it's suffering from anxiety, and this was almost prevented when animals were treated with P50 So it seems that really this confirmed the result we have in vitro, that's showing that amyloid beta is more toxic in the arginine animals than in the proline one. This is only to summarize it. So when we transfer the arginine mice with proline with this P50, then we have healthy neurons. And finally, I want to show you some data we have in a cohort of patients. This is a collaboration with the Hospital of Santiago de Compostela. And we wanted to know whether these polymorphisms were affecting Alzheimer's disease progression. So in a total of 89 patients with mild cognitive impairment, we genotyped them. And this is the condition. This is around 70 years, and it's around 60% females and 40 males. And we did a follow of 4 years. And I show you the result we have with the mini mental, but also other tests has been included with similar results. So as you saw the mini mental, you have this different score ranging from normal to severe cognitive decline. And this is the mini mental at the beginning, the first visit, and this is the mini mental at the final visit, 3 or 4 years later. So as you can see here, there is a worsening, there is a decrease in mini mental score only in the arginine genotype. Only patient harboring the arginine genotype worsen. And this is the deterioration, that is the difference, the percentage of deterioration of cognition, just measured by this, but mini mental, as you can see here, the arginine, the deterioration was higher than in patient harboring the proline allele. So we can see a quite nice relation between the arginine and a faster progression of Alzheimer's disease. Here is another way to express this. This is also the final diagnosis. So as you can see here, the patient with proline allele didn't develop much Alzheimer's disease. And only patient with arginine developed either prodromal or dementia Alzheimer's disease. We also, this correlates very nicely also with phospho-tau. We have higher level in the arginine patients. And we are now doing experiments in vitro, and this is the neurons, proline and arginine neuron exposed to amyloid beta, oligomerized amyloid beta. As you can see here, there is an increase of phospho-tau accumulation induced by amyloid beta in the arginine neurons. You have here the quantification. Then, as a conclusion, we demonstrate that P53 is really involved in neurotoxicity caused by oligomerized amyloid beta, and we established that there is important the crosstalk between the mitochondria and the nucleus in this neurotoxicity. So when we identify indeed a polymorphism that when express the arginine variant, this